Good evening friends, you are most welcome to Think Eyes channel. Today we are going to discuss Practicing Anthropology by MAAN IGNOU, and our topic is Advanced Areas in Practicing Anthropology, in which we are going to discuss hemoglobin variants. Let us begin the topic. Besides ABO blood groups, more than 200 structural variants of human hemoglobin are reported, but only 3 of them, HbS, sickle cell trait, HbE, HbC, are found in fairly large areas of the world with heterozygote frequencies of about 10% or higher. The Khmer populations of northern Cambodia and adjacent areas of northeastern Thailand in Southeast Asia show a highest concentration of HbE 55.2%. This hemoglobin variant HbE has been found in high frequencies in eastern India among the Ahom, Khasi, Assamese, and Totos, among whom it ranges from 20% to 58%. In comparison to HbE, the study of on HbS or sickle cell hemoglobin has been carried out much more extensively in India. It is well established that the HbS gene is widely distributed in India except in the eastern region, particularly Bengal and Assam. It could also be noted that sickle cell gene is mostly found among the tribal population. A few other variants, HbD in the northeast of the Indian subcontinent, HBO in the Indonesia in Celebes, attain heterozygote frequency 5%. However, a great majority of HB variants are rare and do not reach a gene frequency of 1%. Balanced polymorphism. Evidence is now strong that many polymorphisms are maintained in human populations by balancing selection. Such a balance is seen in sickle cell anemia, which is found mostly in tropical populations in Africa and India. An individual homozygous for the recessive sickle hemoglobin HbS has a shorter expectancy of life, whereas the life expectancy of the standard hemoglobin HbA homozygote and also the heterozygote is normal. Though heterozygote individuals will suffer periodic problems, the sickle cell variant survives in the population because the heterozygote is resistant to malaria and the malarial parasite kills a huge number of people each year. This is balancing selection or genetic polymorphism, balance between fierce selection against homozygous sickle cell sufferers and selection against the standard HGBA homozygotes by malaria. The heterozygote has a permanent advantage, a higher fitness so long as malarial existence, and it has existed as a human parasite for a long time. Because heterozygote survives, so does the HGBS allele survive at a rate much higher than the mutation rate. Lactose tolerance or intolerance. There is considerable interest today in investigating genetic base of difference in human metabolism, energy, and material transformation within cells as those differences interact with culture and health. Lactose intolerance and alcoholism are to be mentioned in this regard. The ability to metabolize lactose, a sugar found in milk and other dairy products, is a prominent dimorphism that has been linked to recent human evolution. G6PD, glucose 6 phosphate dehydrogenase. Glucose 6 phosphate dehydrogenase human polymorphism is also implicated in malarial resistance. G6PD alleles with reduced activity are maintained at a high level in endemic malarial regions. Despite reduced general viability, variant A with 85% activity reaches 40% in sub-Sahara Africa but is generally less than 1% outside Africa and Middle East. Genetic variability of major histocompatibility complex MHC in human populations. Human major histocompatibility complex MHC Molecules are called human leukocyte antigens HLA, in short the genetic control of the body's immune system, which were discovered in 1958, Dorset 1958, and is located on chromosome 6. It is well known that several HLA genes exhibit an extremely high degree of polymorphism. In particular, the HLA-A, HLA-B, and HLA-C gene in class 1 and the HLA-DR and HLA-DQ genes in class 2 are highly polymorphic in various ethnic groups. Thus, these genes are very useful for tracing an evolutionary history of human population. The genes of the major histocompatibility complex MHC are highly polymorphic, and this diversity plays a very important role in resistance to pathogens. This is the type of genetic variability involved in acceptance or rejection of organ transplant, defense against cancer, and resistance to disease such as malaria and measles. DNA polymorphism STR and SNP. The study of protein polymorphism has indicated that the extent of genetic variation in natural population is enormous. However, the total amount of genetic variation cannot be known unless it is studied at the DNA level. The results so far obtained indicate that the extent of DNA polymorphism is far greater than that of protein polymorphism. What is DNA polymorphism? Non-coding nucleotide sequences that contain base pair variation that do not appear to have a phenotypic effect on the individual are referred to as human DNA polymorphism. A DNA polymorphism is any DNA variant recognizable by a change in DNA sequence that occurs with a frequency of greater than 1%. Mutation change nucleotides over the course of evolution. Other evolutionary forces such as natural selection and random genetic drift determine the fate of new mutation. Most mutations are eliminated from population. Some mutations at a particular DNA site reach polymorphic frequencies, that is over 5% of the individuals in the population carrying the mutation, and are called polymorphic DNA site. Recognizable DNA polymorphisms or marker DNAs are now being used to locate genes that cause genetic diseases. The DNA sequence in the human genome comprises a string of approximately 3 billion nucleotides, which is packaged as substrings in the haploid set of 23 chromosomes. DNA is not only present in the nuclear region of the cells but are also present in the mitochondria of the human cells, known as mitochondrial DNA. mtDNA comprises 16,569 nucleotides. Mitochondrial DNA are transmitted only through the females, hence both the males and females receive their mtDNA, mitochondrial DNA, from their mothers only. The hypervariable region of the mtDNA, mitochondrial DNA, has 5 to 10 times greater mutation rate than nuclear DNA. Mitochondrial DNA, mtDNA, is present in a high copy number and can be recovered from skeletal remains, hair shaft, etc., which are poor sources of nuclear DNA. DNA polymorphism versus biochemical polymorphism. What are the advantages in population genetics? Traditionally, population genetics studies have used blood group, red cell enzymes, and serum protein polymorphism. Such polymorphisms are generally in the functional region of the genome and therefore under effects of the natural selection. This leads to restricted level of variation. Further, not all polymorphisms can be detected using the traditional star gel electrophoresis technique. DNA polymorphisms, especially those that are in the non-functional regions of the genome, are neutral polymorphism and therefore exhibit much greater levels of variation. This property makes DNA polymorphism extremely useful for population genetic studies. Further, because at the DNA level one can detect a much greater number of polymorphisms it has become easy to pick and choose polymorphisms in any region of the genome. This has facilitated the reconstruction of heliotype and the study of haplotype variation, which is far more informative for population genetic studies. How is DNA polymorphism detected? The simplest method of detect known single nucleotide DNA polymorphisms by use of restriction and nuclease. Such polymorphisms are called restriction fragment length polymorphism RFLPs. Fragments of different length produced by the restriction enzyme can be distinguished by the altered morbidity, the altered mobility of the restriction fragments on gel electrophoresis. These variations in the nucleotide sequence are not expressed phenotypically because these variations are due to sequence differences in the non-coding region of the genome. The gold standard is, of course, DNA sequencing. DNA polymorphism detection has been greatly facilitated by the invention of the polymerase chain reaction PCR technique, which requires nanogram quantities of DNA. In early 1980s, several groups of molecular biologists began to map the human genome using restriction fragment length polymorphism RFLPs.